Let's talk about the genetic code. Now remember, the genetic code and really this whole unit revolves around protein synthesis. That is, how do we get from the DNA, or how does the DNA cause the making of specific proteins? And there was a two-step process, of course, and the first step was called transcription, and that involved taking the DNA in the nucleus and using it to make mRNA, so a strand of RNA that acts as messenger RNA. And step number two is called translation, and that involves taking the mRNA that I made in the first step and using that to direct the synthesis of a protein. And that's really what we're going to be talking about here, is translation, and specifically, like I said, the genetic code. Now the genetic code is basically this: every possible combination of A's, T's, A's, U's, G's, and C's, and what amino acids they code for. Because let's remember, we're talking about protein synthesis here. And what's a protein made of? A protein is a chain of amino acids. So to make a protein, I have to basically string together a bunch of amino acids. And somehow the DNA is directing which amino acids go where. And it does that, first of all, through the first step, transcription, which we talked about, in which case we're making a strand of RNA, of mRNA. And translation is the second step, and that is taking that strand of RNA and figuring out what amino acids it codes for to make what protein. So we're just going to make up a string of amino acids here. So I'm just going to say we're in the middle, and let's just make up some RNA. So put some codons there: A, U, G, C, 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 A, U, let's see, and maybe, I don't know, a G, A, C. Okay, just make up a few. Okay, so there's a reason I drew them in groups of three. We found out, or scientists have discovered, that every group of 3 nucleotides is called a codon. And every codon codes for 1 amino acid. So I've got on my board here, I've got 4 codons, so that would code for 4 amino acids. Now scientists have put a lot of work into figuring out every possible combination of A's, G's, U's, and C's and what amino acid they code for. And this is it. This is a summary of their work. This is the genetic code. This is a table that tells you every possible combination of bases and what amino acids they code for. Alright, so let's figure out how this works. The first codon was AUG. Okay, so what amino acid does that code for? Well, the first letter is A, so I'll go first letter, find A. The second letter is U, so I go to where A and U meet, and I find it, AUG, and I see that that codes for this amino acid, methionine. Okay, the next one, CCC. Go C, C, go to where they meet, CCC, that is proline. Okay, the next codon was AAU. First letter A, second letter A, go to where they meet, and I see AAU codes for asparagine. And there are abbreviations for these, but you don't need to know them. And the last codon we have here is GAC. So G, find A, go to where they meet, and C, and I see that it's aspartic acid. Okay, so that short little section of RNA that I wrote down codes for a chain of amino acids consisting of those 4 amino acids. Now, a couple of things about the genetic code here. First of all, you would have noticed when I looked at the first amino acid, the first codon, AUG, it says this is the initiation codon, or the start codon. Now remember, the thing that actually makes the protein is called a ribosome. Now a ribosome doesn't just find the RNA and start making it right from the start. In fact, what happens is the ribosome is going to zip along the RNA. It won't actually start making the protein until it sees that codon, AUG. So it's like a start sign for the ribosome, telling it, start making the protein here. And of course, if there's a start sign, there has to be a stop sign. In fact, there's 3 possible stop signs, UAA, UAG, and UGA. So when the ribosome sees those codons, it knows protein is done, finish up here, release the protein, it's all good. Okay? Now, there's 3 important characteristics about the genetic code. The first one is that the genetic code is continuous. And what that means is every group of codons is in a sequence. It's just like reading words on a page. So the first codon will code for an amino acid, and then there's no punctuation or spaces. The very next codon will code for the next amino acid. So it reads like a sentence from one end to the other. Okay, no punctuation, no spaces, anything like that. So that's what I mean when I say the genetic code is continuous. The second characteristic of the genetic code is that it's universal. Now what does that mean? It means that no matter what living thing we're talking about, this genetic code is the same. What I mean is serine, so these codons code for serine, whether we're talking human beings, whether we're talking bacteria, whether we're talking cats, dogs, any living thing. Okay, this is the same for all living things. So the genetic code is universal. The last characteristic of the genetic code I want to talk about is that the genetic code is redundant. Okay, now what does that mean? Well, notice that in a lot of cases, if you take a look at proline, there's several amino acids, in fact almost all amino acids are coded for by more than one possible codon. And that's what redundancy is.